Okay, well, welcome everyone to the 2025 Wheatstone lecture. So we have typically a few words at the start just to explain the context of the Wheatstone lecture and who Charles Wheatstone actually was. So he was born in Barnwood, Gloucester in 1802, and the family moved to London in 1806. And his father made and taught music and musical instruments and music, respectively. So he quickly became fascinated by the wonders of sound and transmission, and I think there's quite often that analogy between science and music, and became quite the experimentalist. And he published his first paper in 1823, so he was 21. And before long, he began to make his name making various inventions, such as the English concertina. So he came here to King's in 1834 as a professor of experimental philosophy. And in 1836 was granted permission to have a lab. Very lucky man. And what we would say today, he equipped his lab and conducted experiments in electricity. And in 1837, he and Cooke patented the electric telegraph. And Cooke brought the business expertise and Wheatstone was the technical know-how. So the company was registered in 1845 and Wheatstone received £33,000, but that's well over 4 million in today's money. So he did quite well out of that. So this is the wind fiddle, which looks like it did not succeed, but the concertina, I think, is a bit more familiar. So so here is his telegraph, sorry. So he invented this Playfair cipher, which was one of the first ciphers to encrypt pairs of letters. So he was also interested in secrecy, so this was through his work in telegraphy. So initially it was rejected by the Foreign Office because they thought it was too complicated. So he offered to demonstrate that most boys in a nearby school could learn to use it in 15 minutes, and at which point the Undersecretary responded, "That is quite possible, but you could never teach it to attachés." So that's a statement on the government, I guess. So amusingly, although he was an attraction and obviously an asset to King's, his lectures were something of a disaster. So he was very clear and committed, enthusiastic in conversation, but in front of an audience, he would resort to mumbling and face the board. So no TEF in the 1830s. However, he was a prolific inventor and a really instinctive understanding of what would work and also how to make things better. So this, of course, is what he's best known for, is the Wheatstone Bridge. He didn't actually invent it, but he made it into the format, that is the most efficient. So one thing he did invent as a result of his developing interest in optics was the stereoscope in 1838. And it turned out to be one of the most popular toys of the 19th century. So, and of course, I guess a precursor to our 3D films of today. So I think this brings me neatly to our current event, our current Wheatstone lecturer, Professor Hagan Bayley, and Sergey is going to introduce.
Thank you. So, a very distinctive trait of Wheatstone, as Ruth just said, is that he was really talented at applying fundamental knowledge to engineer practical and useful instruments that have been crucial in the development of physics, engineering, and society in general, such as the Wheatstone Bridge or the telegraph. And this interest in combining different fields of knowledge and looking for direct practical applications is something that underpins the research path of our lecturer tonight, which is Professor Hagan Bayley from the University of Oxford. Hagan combines protein chemistry, molecular genetics, biophysics, and cell biology to explore the nooks and crannies of membrane proteins, and in particular channels and pores. His lab investigates both the fundamental properties of these proteins and their applications in biotechnology. And more precisely, Hagan Bayley's research focuses on the development of engineered hemolyzing nanopores for stochastic detection, including single molecule covalent chemistry and ultra-rapid biopolymer sequencing. More recently, his lab has developed techniques for the fabrication of 3D tissues, both living and synthetic. In 2005, Hagan founded Oxford Nanopore, which has manufactured the portable Minion and other DNA and RNA sequencers, which, as you all know, are really successful and a very beautiful example of how lab discoveries and fundamental discoveries can have a direct and useful impact for society. Hagan graduated in chemistry from Balliol College in Oxford in 1974. He then moved to Harvard to obtain his PhD in chemistry. And after his postdoc in MIT, he started his academic career at Columbia University, then moved to University of Massachusetts Medical Center, and finally to Texas A&M, where he was the professor and head of the Department of Medical Biochemistry and Genetics. In 2003, he moved back home as professor of chemical biology at Oxford. Hagan has made many, many, many discoveries during his scientific career. He has pioneered and developed nanopore field for sequencing, has more recently shown how to fabricate 3D tissues through droplet assembly, and he has held all possible grants, and his work is continuously published in the best possible journals. He amasses over 50,000 citations, and it's not a surprise that his work has been recognized with all possible awards and merits, and the list never ends, right? So, for example, in 2009, he was elected the Chemistry World Entrepreneur of the Year. In 2011, he was elected Fellow of the Royal Society. In 2012, he was awarded the Royal Society of Chemistry's Interdisciplinary Prize. 2017, the Monolos Medal for the Learned Society of Wales. And in 2019, the Mullard Award of the Royal Society. In 2018, he held the Kavli Chair for the Delft University of Technology, and he's currently the lead researcher for the Oxford Martin School. On a more personal note, I have to say that Hagan is simply astonishing and a brilliant mind. Those that work with single molecules, we really like rare events. And so, well, Hagan is a rara avis in the wood sense. With someone with such a translational spirit, one would expect that he concentrates on the technology development and that the scientific breakthroughs come after. But this is absolutely not true in the case of Hagan. He's a deep thinker, a true chemist, fundamental, that likes to challenge the current views and push them further. Someone in the field told me once that we can't simply wait the same time to publish as Hagan does. And this is true because he just simply goes for excellence, and he's not happy until he has a rounded and finished paper. Quality over quantity. And he's been always an example for me with the difference that he delivers quality in great quantity. So I'm sure that you will appreciate how this extensive and impressive track record, working across disciplines of chemistry, bioengineering, and biology, and provide direct application to fundamental discoveries resonates with the Wheatstone ethos. There's one thing, though, on which Hagan, like many other Wheatstone lecturers in the past, clearly outperforms Wheatstone himself. Apparently, Wheatstone was very shy and a terrible lecturer. So much so that his good friend, Michael Faraday, had to deliver the lectures on his behalf. Hagan, instead, is an excellent and very engaging speaker, and that's why I'm so delighted that he has accepted to deliver our 2025 Wheatstone lecture tonight. So without further ado, please join me in warmly welcoming Professor Hagan Bayley.
Oh, gosh, well, thank you both for the introductions, and yeah, what can I say? I did hear that Wheatstone was an abominable lecturer, so I promised not to turn my back to the audience. But Sergey, would you like to go ahead and give the talk? Or I was also told just before I started that Maxwell lectured in here too, which is a bit intimidating. So I thought what I'd do today is give a rather sort of general descriptive lecture of what you can do with nanopores. If any of you have any difficult technical questions, I can give you the email addresses of people in my group who can answer them. But I'm just going to give the big picture today. So our cells are bounded by membranes, and all the organelles in our cells are also so bounded. Of course, membranous organelles are very trendy at the moment. And these membranes contain proteins, all sorts of proteins that are enzymes, proteins that handle signals, proteins that transport molecules in and out of cells. But one particular class of proteins that is very interesting has become known as nanopores. But these are proteins that make large pores in membranes. Here's one example here of streptolysin from Streptococcus. And these proteins are used by bacteria to attack us, but we also have proteins that we make that we can get our own back on bacteria as well. So these molecules, I think, are very interested in basic science. I'm not going to sort of go into the basic science side of things today, but I hope to persuade you that they're valuable in biotechnology. So just to give you one example, here's our favorite protein. Other membrane pores are available, but this is the alpha-hemolysin protein pore, and this is a toxin that's secreted by Staphylococcus, not Streptococcus in this case, and it's a very simple protein. It's just 293 amino acids. It's secreted as a water-soluble monomeric polypeptide, and it assembles into membranes to form a pore that has 7 subunits, a heptameric transmembrane pore, which you can see in this crystal structure here that was solved quite some years back. So this protein is rather like a mushroom, and all right, did we just lose the sound? Hold on. Try again. Okay. The protein looks rather like a mushroom, and it's the stem of the mushroom that goes through the lipid bilayer. And here's the top view of the protein. The wild-type protein has 7 identical subunits, and you can see it in this actually quite old AFM view here. So just to give you an idea of the scale that we're looking at, a human hair is about 80 microns in diameter. In fact, varies quite a bit from person to person. But the pores that we're looking at are nanometers in diameter. In fact, the hemolysin pore is about 2 nanometers in diameter. So it's about 1/50,000th of the, don't do that, why do you do that? Okay. It's about 1/50,000th of the diameter of a human hair. And that's very useful to us because we'll be using it for sensing and sequencing. So it has about the diameter of some of the molecules that we're trying to look at, as you'll see in a moment. So some time ago, we asked the question of whether protein pores might have useful applications. There's been a lot of work on antibodies and enzymes that have been shown to be useful in biotechnology, but could we do anything with protein pores? And at the time, that was thought to be kind of foolish because at that time, people felt that membrane proteins were very hard to handle. They just turned out to be different to handle, not really hard to handle. So to tackle this, issue, one thing I'd like to emphasize is that we do really multidisciplinary research. So we have a research group that has some chemists in it, some biologists, physicists, some device engineers, and so on. And some of the people in the group work on the basic properties of nanopores, their structure, how they get into membranes, and how they function, and so on. But an increasing number of people in the group really work on their applications. And these range from sensing and sequencing that we'll come on to briefly, single molecule chemistry, and also the ability to build synthetic tissues, which stemmed in a rather oblique way from looking at nanopores. So if there's one measurement that you have to remember, you only have to, for the first half of the talk, it's this one. So we can take a piece of plastic that you can see here, with a little hole in it, and we can make a membrane in that hole from phospholipids, and we can have a salt solution on this exploded view here, on each side of the membrane, and then we can put a tiny amount of protein into one side of it, and we do this in such a way that one of these heptamers inserts into the membrane. Then if we apply a potential, we can measure the current that flows through the protein. That current is tiny compared to the current in a light bulb. It's at the picoamp level, but a picoamp is 6 times 10 to the 6 ions per second, so it's actually quite readily measurable. But the point is that if something happens inside the protein, we can see a change in that current, and that's what allows us to sense molecules and to sequence DNA and RNA. So again, just to recap on the structure, if you entered this pore from the top, it'd be about 3 nanometers in diameter. There's a kind of cavity inside it that's involved in storing this part during assembly, but we won't talk about that today. And then there's this part that goes through the membrane where most of the action will be, and this has a diameter of about 2 nanometers. So the point is, if you want to detect a molecule, it's going to have similar dimensions to this pore. If it was too small, you wouldn't see it. If it was too big, it would block the pore. And similarly, for looking at polymers that you might want to sequence. So I'm going to give you a little bit of history to start with. So one thing that we did some time ago is invent this technique called stochastic sensing, where we can detect single molecules using protein nanopores. So as I said earlier, we just put one copy of a protein into a membrane. We measure the current that's going through that protein, the ionic current. It's not carried by electrons. It'll be carried by, say, potassium and chloride ions. And here we see 2 states of the protein here with something bound and then the empty state of the protein. So if we follow this ionic current, we can watch individual binding events. So a molecule binds, it comes off, binds, it comes off, binds, it comes off. And we can use that to determine the concentration of an analyte in solution, and also, as you'll see later, these, the shape of these, of the current during the binding event allows us to identify the analyte. So here's one very simple example that I like to show, but it also shows that we do quite a bit of protein engineering. In this case, we've taken the pore, we've changed some amino acids inside it, so it will bind a cyclodextrin molecule. Cyclodextrins are water-soluble cyclic oligosaccharides that will bind organic molecules, and we've altered the pore in such a way that this cyclodextrin will bind like an adapter inside it, and that allows us to monitor the concentration of this drug molecule ibuprofen in solution. And this is about 50 milliseconds across, so each of these spikes is a binding event. And interestingly, you can see there are 2 types of binding events, and that's because when you buy this molecule, it's a racemic mixture of R and S forms, and because the protein itself, and indeed the cyclodextrin, is a chiral entity, the binding events form non-covalent diastereomers, which can be distinguished from each other. And this is a 50-50 mixture of the 2. So this just shows the power of the technology. It's rapid, you can look at fairly low concentrations, and you can even distinguish isomers from each other pretty readily. Okay, so based on that, as Sergio was saying, we formed Oxford Nanopore way back in 2005, and initially that company was formed to look at small molecules. So we just wanted to look at biomedical analytes. And it was really an Oxford company, Gordon Sanghera, who's still the CEO, worked with Alan Hill of glucose sensor fame in Oxford, and Spike Wilcox was also an undergrad and did his deep film in Oxford. But pretty soon, after we started the company, we thought at that time, I think it would be pretty hard to do, quite honestly today, to take on something more challenging, this idea that had been around for a long time, that instead of just sensing small molecules, you could run a polymer through the pore, i.e. DNA or RNA, and read off the bases as the polymer went through the pore and do complete genome sequencing and so on. That turned out to be quite difficult to do. You just couldn't add the DNA to the pore and read the bases off. We had to use an enzyme to feed it into the pore. For several reasons, it's much easier to work with double-stranded DNA than single-stranded DNA, and it's single-stranded DNA you want to look at, so we can use an enzyme that will separate the strands, and then the neighboring bases on any particular base affect the current in the pore too, so you have to use quite a complicated algorithm to read off the sequence. Nonetheless, that could be done. It took a long time, so it really took about 7 years until we got some not great sequence, but usable sequence. And that ended up with the company making the so-called MinION. And the interesting features of this is it's a very inexpensive technology. It's portable, as you can see here. And also, it doesn't have the limitations of normal DNA sequencing technologies in that you can keep running large pieces of DNA through the pore. The record is about 4 megabases. If you go through the numbers at about 2 milliseconds a base, you can see it takes a long time to read 4 megabases. Normally, you're looking at about 10,000 bases and so on. And then another feature of this device that we'll come back to later is that you can look at about 500 pores in parallel. So one thing that we're trying to do in the lab is produce low-cost applications, and this and other sequencing methods that other companies have devised has really brought the cost of sequencing a human genome down to way below £1,000. So it's really the cost of most medical tests. So it took 7 years to do, and a little while back I went and opened this drawer in the company to see all the things that had gone wrong during those 7 years. And actually this is the 2012 version here with a bit of blue sticky tape on it. But these were all the kind of original devices in the so-called Nanopore Museum. And it did make me think of Professor Wheatstone with all the amazing things that he invented. And indeed, well, he participated in making the telegraph, as you've heard. The concertina actually is very different from a telegraph, so he had a kind of wide-ranging mind. And then, of course, he was into stereoscope, and there was a lecture a little while back, I think, here with Brian May, right, who collects these things. And his original one actually was before photography, so it worked on sketches. But anyway, it's sort of slightly reminiscent of what we went through. And then, of course, this device has seen lots of applications. Basically, you can sequence viruses very readily. You can sequence things like Ebola virus, Zika virus, and of course, there's no such thing really as Ebola virus. There are hundreds of different Ebola viruses that can be put on a phylogenetic tree, some of which are more deadly than others. And also, you can sequence through modified bases, for example, quite readily now. And it was used during, I think this is the 3rd paper on the completion of the human genome. I think it might be complete at this point, I'm not sure, but this was kind of joining the dots at of the unsequenceable parts of the genome. And nowadays you can get devices that have much higher throughput than the MinION. This Prometheon, which is actually really a collection of MinIONs, has about 300 times the throughput. So, sorry, I'm getting completely discombobulated here. Okay, so this machine can sequence about 10,000 viral genomes in a day, because they're barcoded, so you can distinguish one sequence from the other. So that's a little bit of history. So what are we doing now in this area? Well, there are about 20,000 genes for proteins in the human genome, less than people thought before it was sequenced. That makes, we don't know how many RNA transcripts, 100,000 is probably a very low estimate. But there are millions of different types of proteins in cells. And by that, I mean that the proteins that are made from these genes are modified. So they have post-translational modifications, such as phosphorylation, acetylation, methylation, putting on sugars, all of those kind of things. And then they're often processed in various ways. The RNA that makes them can be spliced. The proteins themselves can be modified by proteases. So one thing that we're trying to do at the moment is use nanopore technology to count the various protein species in a cell, and also to map these modifications within proteins. And we'd like to bring this, remember, a single molecule technique, so we'd like to bring this down to the single cell level, which I think is actually impossible. I'll tell you why later. Okay, so one problem is that proteins won't go through nanopores, right? So they're not negatively charged, uniformly negatively charged like DNA, and there aren't any enzymes that really drive them through one amino acid at a time. So the first thing that we did was use the so-called process of electroosmosis to drive the protein through the cell, so through the nanopore. So what do we mean by that? So let's see, that's one thing. That's going to go on to the PDF. Yeah, so basically what we do is take the protein pore, modify it so it becomes quite highly ion selective, so that when you apply a potential, in this case, for example, negative ions are moving in one direction, and each of those ions will take with it maybe 10 waters reasonably tightly bound. So there's a big flow of solvent through the pore, and it turns out that this exerts enough force on the protein to unfold it and pull it directionally through the pore. And we've been working on protein concatenamers mainly that were provided by Sergei's lab, and I'll just show you, I can tell you more details afterwards if you want to hear them, but just basically go over the kind of our preliminary results that we've had over the last year or so. So basically, if we take a protein, we can modify it with the kind of modifications that you will see in a cell. So we can phosphorylate it on specific residues. We can add peptides to it like glutathione, that's a known post-translational modification, or we can attach sugars, in this case a trisaccharide, to the protein. And without going through the data, you can actually see these as they appear in the pore. And one thing that is very important is that we can look at intact proteins. So a lot of other groups are looking at digested proteins, but we can get intact proteins to go through the pore. So in this particular case I'm showing on top, this protein is over 1,000 amino acids long, and we can identify a phosphorylation site in the middle of the protein. And here's another one that's about 700 amino acids, and the point here is that we can identify 2 phosphorylation sites that are almost 300 amino acids apart, and that's actually quite hard to do by mass spec, for example. There are many different, this is something that comes up again and again with nanopores, there are many different post-translational modifications. So at the surface of things, it looks like it might be impossible to use this technique with real extracts from cells. So we know that there are about 400 so-called PTMs, but it turns out there are about 13 highly abundant ones that make up over 99% of the PTMs. And we can identify most of these, but it's still quite problematic to distinguish all of them in a single run through the pore. So just to give you an example of that, we've been trying to distinguish this tripeptide attached to the pore from a phosphorylation site. That's not always possible. Sometimes they, depending where they are in the protein, they will give the same current level. And one thing that we've been doing recently is using binders to distinguish different PTMs. So this is just one example here, where we've taken this organic molecule that will bind to phosphate. It's got 2 zinc atoms in it that ligate to the oxygen on the phosphate. And you can see here that this signal has now been changed to a very noisy signal. And to cut a long story short, we believe it's, once this is bound, these bonds are opening and closing, producing this signal, which provides the additional information you need to distinguish these 2 modifications from each other. Okay, so just for this part, we've basically got very long proteins to go through pores. Average length of a protein in the cell is about 400 amino acids. We can detect post-translation modifications that are really quite far apart, and we can target modifications using various different binders. So now we want to move on to look at real proteins and use arrays with different pores in that I'll come to in a little while in another context. One interesting thing is that in a human cell, your average cell, there are about 10 to the 10 polypeptides. So if you work out the number of amino acids that are in the polypeptides compared to the number of nucleobases in the genome, there are about 1,000 times more amino acids than there are nucleobases in the genome. So it's to even look at all the proteins in a single human cell, I think is going to be very difficult until someone comes up with a microsecond per base instead of, say, a millisecond per base. Okay, so a second thing that we've been doing with nanopores is going back, almost turning full circle, to look at small molecules. And there's been a lot of interest on and off for a long time in the volatile molecules that we have in our breath. And it's believed that these may be different in people who are sick in various ways. And the way that's looked at now is to collect these molecules in various ways and then put them into a mass spectrometer, or better, a GCMS, to try and identify them. And obviously this is expensive, and this is a very sort of bulky machine. And a second problem is that there are a huge number of volatiles in the human breath, probably more than 2,000. So we've been trying to devise ways to use the a MinION-like device to look at volatile molecules. And I think a very valuable trick that we have is to use covalent detection. So what we're doing is looking at reactive molecules in the breath that will react with a pore for a short time. If they reacted for a long time, it would be fairly useless. We need them to turn over in the pore, and then distinguish a certain subset of molecules. So the idea is that the one I'm going to show you now is we've been looking at aldehydes, and the humans release less than 100 aldehydes, some of which are much more prevalent than others, and we're going to detect those by reversible covalent chemistry, which means that we're only looking at, say, 5 or 6% of the molecules in the breath and ignoring the other ones. It would be disastrous to be looking at all of these simultaneously. And again, just to show you how this works and show you some data, so we've got a pore, and it's got a cysteine residue in it. This cysteine has a side chain that has a thiol group on it, so it's a good nucleophile. This nucleophile will react with an aldehyde to form this adduct that's called a hemithioacetal. And we'll come back to this in a second. There are actually two different optical isomers that are formed. So we'll come to, that can help us identify molecules. We'll come back in a second. So typically what you'll see, this is putting propanal, the aldehyde propane, in the pore, and you can see these binding events. And these binding events last less than a second. These compounds are very unstable. And the amazing thing is that you can distinguish a whole series of aldehydes that just differ by one CH2 group. So it's hard for me to see from here, but starting with the shortest one over here, getting longer and longer, and you can see that the amplitude of the current block, maybe not surprisingly, gets longer with as you add one CH2 to these chains. And so typically if you put in a mixture, even by eye, you don't have to use machine learning or whatever to get the result out. You can just see by eye that you can distinguish all the different aliphatic aldehydes. Also, we do, as I mentioned earlier, we do quite a lot of protein engineering, and it's semi-rational, I would say, but in this case we thought, what if we put the sulfhydryl group in a narrower part of the transmembrane pore, and indeed that has an amazing effect. So in this case, you can see here the two different diastereomers, the two different optical isomers that I told you about earlier, and they're very closely spaced when you look at the thiol at position 115. But if you move to, gosh, come on, which way are we going here? Let's go back. If you move to 137, these things are really relatively easy to distinguish. And the point of that is that you want to have as much information as possible in your binding events to be able to be sure what molecules you're looking at. Okay, and then finally, we're trying to build new chips based on the Oxford Nanopore technology, where we can have many different proteins. You could have 115 and 137 on the same chip that will make these into very powerful detection devices. Okay, so for this part of the talk, I'm going to speed up, I think, after this, but I think in our lab and other labs and Oxford Nanopore, we've been able to look at DNA, RNA, polypeptides, and small molecules. And this was a picture someone sent me from the Bishop Museum in Honolulu, where they have the tricorder from Star Trek on one side and the MinION on the other side. And if you want to go on the web now and buy one of the original tricorders, it's going to cost you about £100,000. These are very, collector's items. So I would say this is smaller, cheaper, and a lot easier to operate. A lot of buttons on this, which, as Sergi knows, when I've got hundreds of buttons, things can go badly wrong. So, yeah, we're slowly sort of catching up with the Spark and Co. All right, second part of the talk, I'm going to tell you about designer tissues. I said I'm just going over these things at a really quite superficial level, and you can ask me more afterwards. So one thing that we've been doing is making so-called synthetic tissues, and by that I mean materials that have properties that are like tissues. We've been using 3D printing to do that, and we've also moved on to use 3D printing to print living cells. And then one thing we'd like to do is bring these different constructs together to do all sorts of things, as you'll see. And then why do we want to do that? So that the synthetic tissues could be useful as tissue substitutes, maybe. They can be used for drug delivery and so on. And the living cells can be used to screen therapeutic agents. So, for example, we can make little microtumors that are just a millimeter across, put them into the 96-well dish, and then screen them with different agents, and we could do that on a personalized level. Use microtumors made from a single person's own tumor. Ultimately, we'd like to use these to repair damaged organs, and more speculatively, if you watch too many TED Talks, we'd like to use them for organ fabrication and so on. And also you can operate, if someone is doing surgery, you can actually operate 3D printer, obviously from a distance as well, so there are all sorts of far more speculative things you can do. So this idea really arose, as many things in science, complete serendipity. So we wanted to decrease the volumes that we were using for single-channel recordings, because some of the proteins we were using are very rare, and some of the reagents are very expensive. So instead of using that device that I showed you earlier, which can have anything up to a mil of electrolyte on each side, you can get down to 10 microliters if you try hard, we decided to use droplets in oil to do this. So these are 2 nanoliter-sized droplets, and there's lipid in the oil, so it forms a monolayer around each droplet. When you bring the 2 droplets together, a bilayer will form at the interface. And you can put a tiny amount of protein into 1 droplet, wait till 1 goes into the membrane, and then record the current through it. These are 2 silver chloride electrodes here. So that's how this kind of arose. And then we thought, gosh, if we put many of these together, either in 2 dimensions or 3 dimensions, this would be rather like a tissue. It wouldn't have 2 bilayers between each cell, as you have in tissues. It had 1 bilayer. That's actually very convenient, because then we can get these different compartments to talk to each other just by putting pores or ion channels into selected droplets. So basically, we aim to build these droplets into tissues, 2 major types of tissues that you'll come across, where one that has cells that are in extracellular matrix, and they communicate by secreting things, but there are other tissues where cells are quite tightly packed, with gap junctions between them and so on. So it's this kind of thing that we're after. So basically, again, this is what I'm talking about: oil with a lipid in, put 2 droplets in, bring them together. You've got a bilayer between them, and if you could bring more of these together, you might be able to make interesting materials. And indeed, we can print these things that you can see at the bottom. You'll see how we do that in a minute. And so the point is that you can print this compartmented material and also pattern it at the same time at reasonably high resolution, as you can see here. So basically, to do that, some years ago, we developed a 3D printer, and that prints small droplets. These droplets are about 50 picoliters in volume, and so it's very different to a lot of other tissue printing that you'll see that's more like extrusion printing. It's sort of more like squeezing toothpaste out of a tube, but these are individual droplets, and that's how we can get this high resolution. And it's just an example of some of the things that we can print, and as I emphasized, they just have 1 bilayer between each compartment. And interesting, I sort of draw them as circles, but if you think about the geometry of these things, it's actually quite interesting, quite formidable in some ways. But these, each of these compartments, once they're packed, are dodecahedra. And we printed some material here and then photopolymerized it and broke a piece off and put it in the microscope, and you can see the structure of each compartment here. And I thought that was really unique until I found out that the Romans had been there first. And gosh, it's less than a year ago now, I saw this on the web. And there are 33 of these dodecahedra that have been found in Britain. All I can say is we can print a lot more, and they're a lot smaller. Okay, so what can we do with these materials? We can get them to change shape, we can get them to make things inside them, we can get them to transmit electrical signals, we can make them into ionotronic components, so by analogy with electronics, but we're using ions, and we can use them to deliver drugs. So just very quickly, here was our really first effort at getting a shape change by using droplets that contain different salt concentrations, so water will diffuse from one to the other and actually cause shape changes in either 2 or 3 dimensions. Later we moved on to using so-called smart polymers that will contract or expand depending on the temperature. So we could print a little worm like this and get it to shrink, go through an aperture, swell again, go through an aperture, and so on. And in this way, we can get these creatures to go around mazes. So in this case, we've got one of these things that goes in, it curls up around something that we want to transport, and then we take it around the maze. I'm having great difficulty seeing this from here, but yeah, I'm not doing too badly. And then we drop it off at a certain point, and then we can get our creature to come out of the maze in the end. We can also make things inside these droplets. So this is one example where Mike Booth, who's now at UCL, made a light activatable promoter on a piece of DNA. And I won't go into the details, I can tell you after, but when you shine light on it, it will then transcribe, it will clear the promoter, and then you can transcribe that into RNA and make the RNA into protein. So here's one where we've just taken a piece of this printed material in 3D and then shone light on it, and then only certain compartments make this yellow fluorescent protein, the other compartments don't make it. And that can be quite useful because we can turn on electrical properties of these materials. So if we put the hemolysin pore, our friend that you saw earlier, on into the compartments that contain green dye here, we can bring up 2 electrodes and a current will flow between the electrodes. And this really, it's a different mechanism, but it really mimics a neuron in this sort of very fast and directional electrical signaling. But by using our light-activated promoter, we can actually turn that process on. So if we shine light on a construct that contains the hemolysin gene, and we can get directional electrical signaling through this material. Another thing we've got, and I go very quickly through this, but another thing that we're really interested in at the moment is this so-called ionotronics, so using these materials as electrical components. So we can make so-called artificial nerves from hydrogels and then put proteins into them to inject charge into them and signal through them. And in this particular case, we've got, I can't see how many, but about 7 of these packed into an elastomer, and you can send different signals down each one. So it's really quite a powerful signaling method. Then we put droplets together to make very small batteries. This is a concentration cell here, and here's probably the world's smallest lithium-ion, soft lithium-ion droplet battery. And then more recently, we've made much more sophisticated electronic components, and basically that's based on using hydrogels that are either negatively or positively charged, of course they have counterions, but you can use a very expensive micropipette to bring these things together in various ways. So you can, to cut quite a long story short, you can make diodes, transistors, and synthetic synapses and things using this droplet technology. And then you can bring these things together to make logic gates of various sorts. So a simple one on the left side, a NOT gate, an AND gate, and then this more sophisticated, that's an AND, this is the OR gate here on this side. So obviously one of the things that we'd like to do eventually is build these into synthetic materials and then maybe eventually into living materials. And we can already, if you go to this paper, if you want any details, we built a little device that can measure the beating of cardiomyocytes. So in this case, we have this pink stuff is a layer of cardiomyocytes generated from the human stem cells, and we can pick up the signal using this very, very tiny device, as you can see here. So those are synthetic materials. So what I mean by that is these are materials that have some of the properties, or maybe even different properties, from tissues in the body. We can also print living cells. This is where we sort of migrate away from nanopores, really. And I think this is a very exciting area, given all the things that are happening with stem cells, genome-edited cells, and personalized medicine. We can do really these things so quickly, we can use individual cells in this area. And I would say that you see a lot about organoids. Organoids are quite really interesting, but quite problematic in some ways, in that we don't know all the cells that are in them, and each organoid differs from the next. You'll see a pretty picture of an organoid in nature, but the other 95 in the other wells, each one looked different. But by using 3D printing, you can make these tissues in a reproducible way. So I'm running out of time, but just very quickly, you can print the cells, and we print them in the same way. So when we pack them into droplets, you can get down to single cell resolution, but usually we'll have 30 or 40 cells in a droplet. And we print them. When they're printed, they will have this lipid bilayer that I told you about around the outside. But after a couple of days in culture, they actually conveniently eat the lipid, which is shown with this red dye here, so it's gone. And we get cells that are, so this lipid encapsulation helps you get the patterning of the materials, holds them together in a certain way, and which you can't do if you just obviously just throw them all into a tube. And normally, say, well, in particular with neurons, we'll print progenitor cells, so we'll print the cells before they have long axons and so on. But you can see that after they're printed, they will continue to mature or differentiate. So these are cells on day 4 after printing, whatever it says up there, and then day 14 they're beginning to look more like neurons. And then we've been able, with our collaborators, to take these cells and print them into different structures. Here's one that contains, the human cerebral cortex contains 6 layers. This is just 2 layers in this case. And we've implanted these human cells into a mouse brain slice, and it survives, and the cells start sending out processes from the human tissue to the mouse tissue, and using calcium imaging, we can watch even the the mouse and human cells communicate with each other. Eventually, we'd like to make hybrid tissues between our synthetic tissues and living tissues. There are different ways to do that. This is quite an old one of mimicking gap junctions. We have other possibilities at the moment, but we can engineer proteins that will span two membranes, and that will allow us to bring different pieces of tissue together. And ultimately, we'd like to be able to use these materials to repair damaged organs before people need a transplant, for example. Okay, I won't go through all the virtues of synthetic tissues. I'll just finish by emphasizing that always our goal is to make things that are usable. We don't need any more $1 million or $10 million cures for various diseases. So we'd like to reduce the tissue engineering just as the cost of DNA sequencing has been greatly reduced. Tissue engineering right now is very expensive, but we'd like to similarly reduce the cost. So I'll just end by thanking the people in my group, not all of whom worked on these things. They're working on some other things as well. But I'd also like to thank the people who've been in my lab who've taken faculty positions around here now. Stefan Holwicker is a long time ago, is in the audience, but Mike Booth also at UCL, and in particular work I've been talking about, Yuzha Qing, who's now a professor at Oxford, and another Yuzha, one female, one male, Yuzha Zhang, who's did the dropletronic work I told you about at the end, who's now moved to EPFL in Lausanne. So, thanks very much. Thank you.